Okay, so this lecture, I'm going to be starting on the materials on amino acids. So I'll start first of all with the organic chemistry, just describing what amino acids are and what they look like. So I'll start with the groups that make up the amino acids. So we'll start out first of all with this group, NH2. So that's an amine group, hence the name amino acid. That's the amine group. Then we have this group, COOH. This is a carboxylic acid group. You may also see it written as C double bond O and then OH group there, where this here is known as a carbonyl. This is a carbonyl group, but overall it's a carboxylic acid group, and hence the acid in amino acid. And then as well, there's going to be the central carbon that connects all of them. And this central carbon is known as the alpha carbon. That's a denotation that's a biological denotation. It's not so chemical, but in biology we call this carbon the alpha carbon. There's another group that's attached, which is to the central carbon. So these first two are connected to the central carbon. Another one that's connected is just a hydrogen, a standard hydrogen. And then the last group that's connected is known as the R group. And this R group means something. It means something which we will come to later. It's like a wild card. So it just means something. Something gets added on. Typically with an R, it means some kind of alkyl, some kind of carbon chain, and all of the groups that get added on are a carbon chain, something added onto them, different functional groups. But we get to that later on. The first main thing is that how these all come together to make that amino acid. So the way that I like to draw it, or let's take it back and just even more. So let's start off. I said this carbon here, this alpha carbon, is the central carbon. So let's draw that one in first. I'm just going to put a little alpha there. I'm just putting that alpha there just so you guys can recognize it. You don't generally need to draw that, but just so you know, these are the same thing. Okay. Now we can build on the rest of the groups. So let's build on that carboxylic acid group. Let's build on that amine group. Let's build on that hydrogen. Now let's build on that R group. And they all come together like this. And you may see me draw this as this. I do that sometimes just to make it easy. But this is just showing this. This is what it would be written out of as if it was full. Now you can see that there's 4 different groups attached here to this central carbon. And so what we know in organic chemistry, when we see 4 different groups we attached to a carbon, we can say that that carbon has chirality. And that chirality means that there can be an optical isomer, which means that there can be a mirror image form of it. And so what you could do is that you could take a mirror image, and if we have this, for instance, we draw it with our wedge and a conformation. We can make a mirror, the dash and wedge conformation. We can draw this and we can make a mirror image of it, which will change its orientation in space. Now, if we were to, I will just draw that to illustrate the point I'm trying to make. Now, these are the mirror images of each other, and there's that left and the right hand thing that we talk about, where you have the left hand and the right hand. These are mirror images of each other, but they can't be overlaid. And this is important because if, for instance, you have a molecule that needs to be recognized by another molecule as they come together to react, they normally need to have the correct spatial conformation in order to come together, whether that be a ligand and a protein coming together, or a chemical reaction between two smaller molecules. They need to have the correct stereo isomerization, the correct mirror image in order to come together, because my left hand and my right hand are not the same. They won't overlap. We need them to come together to react in an exact conformation. It's not the same, so it won't work. And so these are two different. These are two isomers of the same molecule, and biologically they may interact in a different way. To the extent even where in biology, the stereoisomers, mirror images, are given the notation L and D. And in amino acids, the L amino acid is the one that's seen in biology. And different biological systems, different biological chemical reactions, it will only recognize, especially in animals, will only recognize the L amino acid. This is the one that's seen biologically, this is the one that's seen in, particularly biologically, and the one that's seen in animals. There are some D amino acids in nature. For instance, in bacterial cell walls, they sometimes use D amino acids, but it's the L amino acids that dominate in animal systems, which many of you will be familiar with and may be working with. So we have this L amino acid that we're working with biologically. So the next thing that I need to move on from the structure is to do with these groups here. These groups here, they will, this molecule, as I've drawn it here, wouldn't really exist so much in nature because these groups here are ionizable. They can, ionizable refers to the charge that they may have because things get added on or taken off. And typically, what we see in nature at a balanced pH, or a neutral pH rather, pH something around pH 7, what we would see instead is this. So the amine group's got a positive charge, and the carboxylic acid group has got a negative charge. So to explain what this is and what this means, and to be able to, I need you guys to first understand that this is the structure at pH 7, because these two groups are ionizable, and the point at which they will ionize is at pH 7 is where some an amine group such as this at pH 7 will have an extra proton, will have a positive charge to it, and a carboxylic acid group at pH 7 will lose a proton. See here from their uncharged forms, the carboxylic acid will lose a proton or hydrogen in order to become the charged species as well at pH 7. So that's the pH, or it's going to be 7.5 in cells, but around this pH, this is going to be the form that you will see. Now, I need to explain to you guys why, so that you can predict at different pHs what ionization these groups will be in. And this is also important for when we incorporate the R groups, because many of the R groups have also got chargeable species such as carboxylic acids or such as amines, which may become charged. And this has a lot to do with what the amino acid does and is the function which it imparts onto the protein when it chains together in order to make a protein. So I need you guys to be able to understand, we explained pH and pKa in previous lectures, and to be able to understand the relationship towards pH and pKa on amino acids and what that means downstream. So let's go a little bit into, well, first I keep referring to this as a proton. So let's talk about the reason why. So if we have a hydrogen, and we count the number of protons, neutrons, and electrons, how many of each does it have? So it has an atomic number of 1, means it has 1 proton. It also has a mass number of 1, which means that we've already got 1 mass from the proton, which means so to equal 0, we need to have equal 1, we need to have 0 for the neutron. And then we know that in terms of charge, a proton gives a positive charge, a neutron doesn't give any charge, and an electron gives a negative charge, which means that we need to balance this charge. Electron gives a negative charge, so we can balance it by having 1 electron. Now, let's say that, so this is what the hydrogen is. So let's say that, for instance, the hydrogen lost an electron. Well, what is it then? It's essentially it's a proton. And so what chemists often do, and biochemists as well, they refer to a hydrogen that has lost its electron and thus positively charged. Just refer to it as a proton instead. And rather than hydrogen ions, we just refer to it as a proton, because that's essentially what it is now. So let's look at this phenomenon that I'm talking about of this ionization. So let's start with, for instance, with I'm just going to draw a carbon, and there's other stuff coming off it, but don't worry about it. Don't focus too much on that. Focus instead on this central bit here. So there's going to be something else attached there, but we're just focusing on this little bit here. So let's look at the electrons. Remember the octet rule. Now there's going to be other electrons on the carbon, but one of the bonds which it has with this oxygen, I'm going to draw its electron as a cross there, and it's a covalent bond, so the oxygen is donating 1 as well. And then similarly with the hydrogen, I'm going to draw that as its electron as a cross as well, and the electron coming from the oxygen as a dot. And then the oxygen as well has got 2 lone pairs. So I'll draw those in. So the oxygen in total has got an atomic number of 6, which means that it's got 6. No, that's not correct. Well, it's got the atomic number of 8, which means that it's got 6 electrons in its outermost shell, because it's got 2 in its innermost shell. So it's got an atomic number of 8. 2 electrons are in the innermost shell and they're not shown, but it's outermost shell. Remember that the octet rule means that in the outermost shell, known as the valence shell, these like to have 8 electrons in an alpha shell. So it starts off with 6, donates 1 to the carbon-oxygen bond, donates 1 to the oxygen-hydrogen bond. That's that dot there, and the carbon and the hydrogen respectively donate 1 electron of their own. Hydrogen donates its only electron into the shared bond, which means now that it's got that 2 for its innermost shell. It's only got 1, then it's just that innermost shell that has 2 electrons in it. And then the oxygen's got the 2 lone pairs illustrated here. Now, what can happen is that oxygen is quite greedy, and it's electronegative. It's got quite a pull for electrons, and as such, it's quite happy to take all of the electrons. And hydrogen actually doesn't have quite as strong a pull. It's only got 1 proton pulling on its electrons, so it hasn't got such a strong pull. And so what we find happens is that the electrons from this bond, during the curly arrow coming from the bond, not from the hydrogen, coming from the bond, because it's shared between them. That's the location of the electrons. But those electrons don't believe that belong to either. They're shared. Until what may happen is that the electrons from it may move across to the oxygen, and what you end up with is, and this happens in equilibrium, what you end up with will be the CO negative charge, because it's got that extra electron that's stolen from the shared bond with hydrogen, plus the hydrogen, which is now essentially a proton. This happens in equilibrium. And because it's in equilibrium, it has a constant K, and with the acids, that is going to be Ka. And so what you essentially have is this is HA, this is A minus, and this is of course our H plus, where we're talking about acid-base chemistry and all of that stuff where we're from the previous lecture. And remember, sometimes I call this conjugate base. Well, because this is A negative. It'll be it had a reasonable amount of desire to take on a proton. And something which can take on a proton is known as a base. So this has a reasonable amount of desire to take on a proton. It could take on a proton, but it doesn't really want to, because the Ka in the direction of the equilibrium is such that it's not going to. But it could. It has the capacity to do so, and so it's known as the conjugate base. But it won't if the equilibrium doesn't dictate it to do so. But then this is essentially, like I say, the base. And this group here, this is what I'm describing here with this carboxylic acid. And so when the environment is such that the carboxylic acid will want to give away a proton, it will become charged. Likewise, if we have, for instance, NH2 for the amine group, that nitrogen's got a lone pair of electrons as well. And so that nitrogen can actually take on a proton, because that proton can take on two outer electrons, can take on two electrons to make it to kind of complete its inner shell. Whereas this nitrogen's got a spare pair of electrons that's sitting there with this negative charge. And it's got a kind of a small amount of desire to take on a proton. It's a weak base. And so what you will find is that, again, you can form an equilibrium, and you can get, in an equilibrium, you can get this, NH3. And it's just it's the same thing, but in reverse, because there is a desire, a small amount of desire to take on a proton. And this again as a constant, and that will be a Ka as well. But whereas the Ka for an acid may be quite low because it wants to take on a proton, the Ka for a base will be quite high, because so this will want to donate protons to the Ka will be low. This wants to receive a proton. This will want to give away a proton because that's represented by the Ka being low. This wants to receive a proton. It's represented by the Ka being quite high, and sometimes even represented as being Kb. We didn't really go into that before, and I'm not going to go into that again. But we can represent this on a graph. Different things as well will have a different acids will have a different Ka depending on how much they want to give the proton away. So let's consider, for instance, with an OH group. Let's just consider methanol. Okay, this has just got an alcohol. It's just an alcohol. It's got an alcoholic group, and then that could potentially ionize to form the hydroxylate ion. But it doesn't really like doing this. The Ka is quite high. I just kind of, it's not as high as a base, but it's a fairly high one. It doesn't really want to do that quite so much. Conversely, if we have a carboxylic acid, I'll just draw a general one. Let's say this has got a low Ka, and it's quite happy to donate that proton. The reason being is that the difference between the different states, and when you get the conjugate base at the end, how stable the conjugate base is. So this doesn't happen, or rather, this bit doesn't happen quite so much, because this is unstable. It's not very. It doesn't like that. It's not very stable. It's got this this single electron, and that single electron is flying around. It's very reactive, and so it's in a uncomfortable place, and it doesn't like to be here. So the equilibrium isn't very far on this side. Whereas conversely, this carboxylic acid, it's not strong acid, but it's fairly comfortable to donate its proton. The reason being is that this electron can fly around between this bond, and you have a thing called resonance stability. That has to do with the fact that this double bond here It's double bonds. It's got pi bonds, and the shape of those pi bonds is such that it allows the electron clouds to be spread over that whole area, and it delocalizes the electron. And if you think of the electrons being something somewhat uncomfortable, so this is somewhat uncomfortable, by having the double bond, that allows the electron to kind of get shared into this area, and instead it kind of spreads the discomfort. So you can this is a little bit more stable, so you get a little bit more of it, and this gets reflected. Well, if I move over here now, we can represent this kind of thing on a graph. So the HA, the equilibrium point, and the A plus and the A minus and the H plus, we can represent this the relationship between these on a graph, and this will help to explain why we get the ionized species and at which pH we get the ionized species. To understand that better, to be able to predict it, if I give it to you in a question. So if we were to take this as a graph along the y-axis, let's do percentage ionized. So this is kind of like a also known as ratio of HA to A minus plus H plus, I guess. That's what we've got along on the y-axis. Let me just. There we go. And then on the x-axis on the bottom, let's have pH. Now, if we were to have a carboxylic acid, remember that it quite likes to donate its proton. So let's say that as we increase the pH, let's do pH 1 here, pH 14 here. Say this starts off at a pure solution of carboxylic acid in water, and so let's say it starts off at a pH of 1, some imaginary carboxylic acid. If we were to increase the pH, by increasing pH, that means introducing base, then very rapidly, you'll find that the carboxylic acid will ionize. Ionize very rapidly until it's completely ionized and there's not very much of it left. We don't need this anymore. Keep this up, but we don't need this anymore. Let's even make it a little bit steeper. We'll ionize very rapidly until it's 100% ionized. For example, this is just a thought experiment. I don't think we really see this in reality, but it's just an experiment to communicate an idea. It would, at very low pH, very quickly, it would ionize because the pKa is low. Whereas conversely, let's say the pH 7 is down here in the middle. Conversely, if we were to look at the amine group, which is kind of basic, instead, it wouldn't start, let's say the ionization, increasing base would increase the percentage of ionization around about here instead. Okay, so this is for the reaction of COOH to COO ionized, and this is for the reaction of NH3 to NH2. So the midpoint of which this is 50, remember this is the percentage ionized. The midpoint at which this is 50% ionized. That's 50%. This pKa. Remember, pKa is the point at which something is 50% ionized. So it's the pH is equal the pKa at the point at which something is 50% ionized. pH pKa 50% ionized. And so you can see that carboxylic acid has got a low pKa, whereas an amine has got a high pKa. And if we think of this in terms of an equilibrium, we think of this in terms of our seesaw, which I like to draw for equilibriums. So we have the, let's go with an equilibrium of carboxylic acid to the respective ion. These are an equilibrium, and thus you have a force pushing down on one side and an amount of force pushing down on the other side. But these don't necessarily have to be equal, because the position, if the position of the fulcrum is such that the force being the position of the position of the fulcrum, the bit of part of the T-socks on either side, the position of that dictates how much force needs to go down in order for this to be balanced. Balance doesn't necessarily mean that the amounts on each side are the same. It just means that they even out by the position of the way in which the fulcrum dictates. So, for instance, if the fulcrum was over here, then you would need more force on this side, but they're balanced. They're balanced. They may be flowing back and forth constantly, but the resting point is the amount of force here and more force here, because that's where the fulcrum is. Typically, with something like a carboxylic acid, you have more of the unionized form than you have of the ionized, because this doesn't dissociate as much because it's a weak acid. So the position of the fulcrum is here. So here at this point, if we were to start off at the beginning at a very, very low pH, what does a low pH mean in terms of the hydrogen ion concentration? What does a low pH mean? A low pH means that the concentration is high, because we take the negative log. Remember, the P means negative log. And so when the hydrogen ion concentration goes up, the pH goes down. Because if we have, for instance, times 10 to the negative 7 times 10 to the negative 3, and then times 10 to the negative 1. We're going to take the negative logs of those. That'll be 7, 3, 1. You see that these numbers are actually going up in concentration, whereas these numbers are going down in amount. So a lower pH means more hydrogen ions. Now, if we have a low pH like we do right here, we've got a ton of, we've got more of this. And because we've got more of this, we've got more being pushed down on the seesaw here. Because we've got more pushed down on the seesaw here, we need more of this to balance out. Because we need more of this to balance out, it's not going to go very far over in this direction. It's not going to go in that direction. In fact, it's liable to go in this direction instead, to try and if you've got some of this available, this the A minus, the conjugate base, if you've got any of that available, it's going to want to take up those hydrogens to try and relieve the pressure over here, and then at the same time generating more of this. So we get back to our balanced point again. And so at a low pH, you're going to have less ionization because it's going to be pushing the equilibrium over in this direction because you have more of this. And so it's going to make things out of balance. And so to bring things back into balance, you're going to be removing some of this and generating more of this. So that brings the balance back here. So at a low pH, where pH is less than pKa, you've got a higher amount of this, and it's less ionized. So, for instance, carboxylic acid at a low pH, say pH 1.0, then it's not going to be charged anymore. It's going to be neutral. It's going to be taken on a hydrogen, because there's so many hydrogens in the solution in the system that's going to take some up in order to remove them and generate more of this. For the balance of the equilibrium, this is a 1.0, not 10. It's going to be taking some up. Whereas conversely, with the base, the base is going to want to a neutral pH, because pKa is kind of high. A neutral pH is going to be taking on a proton, so the position of the fulcrum is on the other side. So NH3 plus and NH2. So fulcrum is on the other side, which means that we're going to need more of this. It's going to be more of this. And so if you have any amount of hydrogen ions in a solution, then the amine group is going to want to take them up in order to, it's going to want to reduce this side as much as possible, make this side as much as possible. It's going to be a higher concentration of this, but a lower concentration of this. The position of the fulcrum is moreover on this side, because the position of the pKa is more on this side. However, if we were to add in too much base, remember adding in more base is going to increase the pH. What you're effectively doing, remember, you're measuring the hydrogen ion concentration. And here at pH 14, there's a low hydrogen ion concentration. Here at pH 1, there's a high hydrogen ion concentration. So if we were to introduce base, what you're doing is that you're eating up all of this, these hydrogen ions. You're just eating them up. You add in, for instance, base. But you're going to be eating them up. And so if you introduce base, then you're eating up the hydrogen on this side. And then because of that, there's going to be it's out of balance. Being up the hydrogen on that side, you haven't got as much force there anymore, which means that what's going to happen is that this is going to have to move in the other direction, give up its hydrogen that it had in order to put it onto this side, so you can have some more force being pushed on this side and less force being pushed on this side, in order that they balance out again. To get the ratio back to where the ratio needs to be, the ratio being because of the fulcrum there, it's going to be to get it back to where it needs to be. So it's going to dissociate. So when the pH is high, there are so few hydrogen ions present in the solution that this is going to have to generate some hydrogen ions by dissociating back to its uncharged form, thus having dual effect of increasing NH2 and decreasing NH3 and increasing H plus. to balance out the force again. And so at a high pH, let's say for instance pH of 13, what you're going to have, well, first of all, you're at a high pH, and that pH is above pKa for the carboxylic acid, which is going to be, let's say the pKa is like 2. So you're above 2, which means that this one's going to be charged. And it means that this one, the amine group, now because the pH is high, there's not very many hydrogen ions, protons in solution. You're going to give it away into solution, and you get this instead. This species instead. Now remember that this is dictated by the pKa. pKa 1, pKa 2. And every different group of every chemical, every species is going to have different pKa's. That means for every amino acid, remember there's the 20 protogenic amino acids, which means amino acids which are important in protein synthesis. There's other amino acids that are not used in protein synthesis, so they're non-proteogenic amino acids. But for any amino acid, each carboxylic acid group is going to have a different pKa, and each amine group is going to have a different pKa. But the thing is that I don't want you to. I'm going to give you a question like at pH 13, draw the ionization state, and this is what you would draw. This is exactly what you draw. This is exactly how you answer it, because this one is donated. It's a proton that it quite likes to have. This is donated its proton that it likes to give away, and so it's going to be this would be the answer to that question. But let's go with that. I've completely forgotten where I was going. Oh, each one has its own pKa. And you're thinking, well, that means I've got to remember the 20 proteogenic amino acids. I've got to remember pKa 1 and pKa 2. And that's going to be extremely difficult. So I say, hey, draw at 13. You're like, draw serine at pH 13. You're thinking, well, what's going to be the pKa 1 of serine? What's going to be the pKa 2 of serine? What's going to be the pKa 1 of glutamic acid? What's pKa 2 of glutamic acid? It's going to be really difficult. General thing to remember is that for all the amino acids, the pKa of the carboxylic acid group is about 2. There's a point there as well. It's not showing up in the camera. It's about 2 on all of them. So you don't need to remember all of them. You just need to remember that if the pH that I give you is below 2, then it's going to be the unionized, and if it's above 2, it's going to be ionized. And that relates to this graph which I drew here, because at above pH 2 is greater than 50% ionized, and so we'll draw it like this. Conversely, all of the amine groups, they've actually got a little bit more variation. These are all about 1.9 to 2.1, very, very close together. I mean, it's a logarithmic scale of pH, it's log, negative log, but you know, they're fairly close together. These have got a little bit more variation, somewhere between like 9 and 11. That's you know, 2 orders of magnitude difference, because each one is 10. Each one you go up on the pH scale is times 10 in the concentration of hydrogen ions, because it's log. But you know, it's right about the average. I mean, the ones which are either side, the extremes. There's not many of them. The average is around about 9.5. And I'll make it easy for you guys. I'll give you something like pH 13 or pH 12. So that's way above. So you know, at pH 12 or 13, if I was to give you guys the question, draw this amino acid, or let's draw serine. This is serine, CH2, and then OH for its R group. Draw serine at pH 12, or draw serine at pH 13. This is what you'd have. This would be the answer. for the reason I mentioned. But if I was to say draw it at pH 1 instead, this is what you have. Or finally, if I was to draw it, say, draw it at pH 7. This is what you'd have. Now notice that this structure here has got both a positive charge and a negative charge. We call this a zeta ion. Coming from the German for 2 is Zia, and a lot of the early chemists were Germans. So Zia 2 zeta ion, both charges, 2 charges, a positive and a negative, on the same molecule.